Hi everyone, I'm Chantal,
I'm Evan.
I'm David.
and this week we'll be presenting phenotypic mismatches reveal escape from arms race coevolution by Hannafin et al. So just a few key terms. Coevolution is the reciprocal evolutionary change between interacting species and is driven by natural selection. Reciprocal selection is the selection that occurs in two species due to their interactions with one another and is a critical prerequisite of evolution. And just to define match and mismatch, a match is a locality where sympatric interaction could result in variable fitness outcomes for one or both taxa, and is a locality where coevolution is occurring or could possibly occur. And a mismatch is a locality where distributions of traits at the phenotypic interface are functionally non-overlapping, and it's where there's no coevolution occurring or the possibility of it occurring. And individuals of each species will not experience variable fitness outcomes because extreme traits in one species won't generate ecological effect on the most extreme trait of the other species. So there's a large gap between phenotypes, meaning, for example, there will be little newt toxicity and then large snake resistance. Okay, so just a little background. The newt of the genus Tericha has high levels of TTX, which can be lethal to a variety of predators. So tetrodotoxin is a potent neurotoxin which blocks the propagation of action potentials by binding to the outer pore of the voltage-gated sodium channels in muscle and nerve tissue. And individuals from some populations have enough toxin to kill thousands of mice or up to 10 to 20 humans. And some garter snakes of the genus Thamnophis have evolved resistance to this prey toxin through modifications of the sodium channel structure in skeletal muscle and are capable of ingesting whole adult newts without permanent adverse effects. And the phenotypic interface of the predator-prey interaction between garter snakes and the newts of the genus Tericha revolve around TTX. So snake resistance and newt toxicity do vary geographically. And so garter snakes that are found in areas where there are no newts or non-toxic newts are not resistant to TTX, whereas what the Western garter snake has resistance up to 10 to 1,000 times higher than that of other members of the genus in some population. And this is because population differences in resistance are correlated with functionally important differences in amino acid sequences of skeletal muscle sodium channels. So for the main points, arms races between natural enemies lead to the rapid evolution of extreme traits, high degrees of specialization, and the formation of new species. So researchers examined the variation in phenotypic interface of arms race between garter snake predator and its deadly newt prey to assess the patterns of functional mismatch across the geographic and phenotypic ranges of their interaction. And garter snakes in some populations have evolved dramatic resistance to TTX defense of their local prey. And by evaluating the pattern of mismatches between toxicity and resistance, researchers discovered that predators sometimes escape the arms race through the evolution of extreme resistance, but that prey never come out ahead. And this can depend on the molecular genetic basis of resistance in snakes. So the question that this study was posing is, what is the course of coevolutionary dynamics in this predator-prey system between the garter snakes and the newts? So what do these species look like before coevolution starts? What do they look like while coevolution is underway? And is coevolution ever ending? So for the contribution to science, the evolution of geographic and genetic patterns of variation in newt toxicity is lacking, and data from only a few localities in North America suggest tight matching between prey and predator phenotypes. But a comprehensive survey of newt toxicity has not previously been conducted. So the researchers looked at multiple newt snake subpopulations to theoretically see populations at all stages of a trajectory. And since mutations are random, the length of time required to pass from one stage of a trajectory to another can vary considerably from one population to another. And the researchers were interested in cases where reciprocal selection may have appeared to end on its own because this would contradict the generally accepted models of coevolution.
So for the methods section, this was a descriptive study because there was no manipulated variables. They just took data from the newts and snakes and analyzed it. They had a couple of assumptions they made. The first assumption is that there is no gene flow between the populations, They could test this assumption by doing a molecular phylogenetics to see if there's actually gene flow between the populations. And then the second assumption they made is that they assumed that looking at multiple populations at one time is similar to following just one population for a long period of time over multiple generations. They could test this by actually doing that and seeing how coevolution occurs through generation or generation and see if it matches their data. So they gathered data from newts and snakes from 28 locations across the west coast of North America. So they looked at 383 newts, 2 or 57 at each site, and measured their skin TTX levels using high performance liquid chromatography fluorescent detection. To measure snake resistance, they used 2,449 female garter snakes and injected the snakes with the average TTX levels of their sympatric newts. They chose females because they are larger than males and more likely to prey on newts. We believe it was adequate, but only one loci had a sample of 2 newts, which in our opinion was not large enough sample size to come up with the average toxicity of that newt in that area. Also, in general, a larger sample size would have been better. So the 15, 50, and 85% refers to snake mobility after being injected with their sympatric newt amounts. So what this means is if they were 50% slower, so if they were 50%, that means after they were injected, they could only move 50% of their baseline speed. So the statistical test they used was orthogonal distance, and they also used ArcView GI 3.3 with spatial analysis and post hoc comparison. We are no experts about analysis, but they state that they used a similar approach in a study about arms races between sexes within species. And then this experiment was across species, so it may not be the best method, or it could actually be better. For the results section, what they concluded was that TTX levels varied between new populations. They ran ranges from non-detectable to 4.69 milligrams per newt. They found a correlation between snake resistance and sympatric newts. It was seen TTX levels increased as snake resistance increased. They noted the new population with the highest TTX also had the highest snake resistance. This is what they expected to see, highlighting a coevolutionary relationship. They also found that 1/3 or 10 populations, there was a mismatch between newts and snakes and no coevolution was occurring. These results surprised them, and they learned that it was the case for some newts that had little or no TTX, and then the other case was that snakes had high resistance to TTX. They also found there was no case where the TTX levels was greater than snake resistance levels. The snake was always had the upper hand. So in figure 1, this shows the map of where they took the sample datas. The square represents snake resistance and the circles represent newt TTX levels. So for the circles, for the legend, they just measured how much TTX a newt had. And for the snakes, it was the amount of TTX required for the snake to slow down 50%. All the circled regions are regions where could be mismatch or there is mismatch. The purple regions are caused because there's low TTX levels in newts, and the green regions are caused because of the high snake resistance. So in figure 2, there's A and B, which is also showing similar results to figure 1. As you can see, like prey toxicity and predator resistance are very similar. So that shows a coevolutionary relationship. And then they combine these 2 graphs and put them together to see where the mismatch was occurring. And you can see that the mismatch occurred in places where there was low newt TTX or high snake resistance. In figure 3, this is just another representation of the data. The purple represents where there was low TTX levels in newts, and the green represents high snake resistance. Anywhere between 15 and 85% mobility was considered matched, and there was a coevolutionary relationship. And also this graph shows that there were no places where snakes had could not resist newt TTX, and they were always had the upper hand. Graph 4 shows places like that are mismatched, and this was because of either high snake resistance or low newt TTX. while places that were matched, it's important to note here that Willits has a low TTX level, but there was also low snake resistance, while places like Benton had a high TTX level, but also high snake resistance. So this article does not propose a specific hypothesis or try to falsify other hypotheses. The article's intent is just to explore the dynamic coevolutionary relationship between toxic newts and resistant snakes.
So now we're going to discuss our results and findings. So the scientists, when they did functional evaluation, it showed them that over 1/3 of sample localities represent specific mismatch between predator and prey abilities. In 10 localities, predators can resist selection due to prey toxicity. However, prey are never too toxic to be successfully ingested by local predators. Some populations of predator and prey have not entered the arms race at all. In some cases, predators evolved high resistance to prey toxins, and that is how they skipped the arms race. Mismatches at the phenotypic interface of coevolution. It's possible for predator but not prey to skip reciprocal selection or coevolution in the arms race, which contradicts equilibrium theory and the life dinner principle, which they both state that prey experience stronger selection than predators in an arms race. This means that coevolution should favor the defensive adaptations of the prey over the offensive adaptations of the predator. And by the way, this paper noted that this may be only unique to the newt and garter snake interaction. So obviously other interactions should be studied as well in the future. The adaptive changes in resistance and toxicity are not only influenced by the strength of selection, but also by genetic architecture. For example, much of the variation in TTX resistance in the garter snake results from the expression of TTX resistance sodium channels in the skeletal muscles of the snake. A geographic region that contains phenotypic escalation does not necessarily have to be a coevolutionary hotspot. Some cold spots, which don't have coevolution in them, exist in localities with upper and lower phenotype extremes in both the predator and the prey. Ultimately, coevolution hotspots and cold spots are defined based on the nature of interactions between predator and prey, not the phenotype level. Inferring coevolutionary dynamics. Many factors might enhance reciprocal selection between predator and prey, so it's not possible to be certain that each of the observed localities did demonstrate coevolution. For example, they measured the TTX levels, and 4 populations appear to have engaged in an arms race. However, they also found mismatched localities that appear to never have engaged in an arms race. So because this paper has no hypothesis and it doesn't try to disprove another hypothesis, I would say that the ultimate conclusion that it seems that we still haven't established specific criteria to determine if populations are actually undergoing coevolution or not. The paper still can't tell if certain populations right now are engaging in an arms race or not. So future studies should first try to establish specific criteria to determine if coevolution is undergoing. Also, they should study different predator and prey interactions, not just the snake and the newt. Thank you guys for listening, and see you on Friday.